Good afternoon, and welcome to this session of the 2018 WEP entitled The Forgotten Half of the Brain. Quite an intriguing title. My name is Pierre Magistretti. I am the Dean of the Division of Biological Environmental Sciences and Engineering, and I'm a neurobiologist, and I've been working on this forgotten half of the brain, about which I will tell you more. And the co-speaker today is Professor Yves Agid, whom I introduce now briefly, and he will speak after me. And he's a distinguished neurologist. He was the head of the Department of Neurology at the hospital in Paris, La Salpêtrière, a very famous place for neurology. One of the founding fathers of neurology, Charcot, was a professor there, and since then there has been a very long tradition for studying and trying to cure diseases of the brain. And Yves Agid and myself, we have known each other for a while. He's a clinician, I'm a basic neuroscientist, and eventually, as I like to tease him, he has been studying neurological diseases with the assumption that neurons were the main cause of these diseases. But later, as he progressed in his clinical activity and also in his research, because he was running a large clinical research group, he realized that actually the forgotten half or the other half of the brain was important. And so we started exchanging about ideas on how the basic work that I've been doing on these cells, which I'll tell you more about, and how that could relate to higher brain functions and diseases. And we got together, and over the last couple of years, here and there, when we have some free time, which is not very often, but we managed to write a book which will come out, will be published next month in Paris, a publishing house which is actually well known to publish books that are for the general public, in which important issues about science and technology are presented. The publishing house is called Odile Jacob, Édition Odile Jacob. The French-speaking people here, I'm sure, know about. Often these books are translated, so I hope one day ours will also be translated in English. But let's start with what do we mean with the forgotten half of the brain. If I ask you, which are the cells that make the brain, that make up the brain? I'm sure everybody will answer. Neurons, okay? And you're right. Neurons indeed are important cells in the brain. You have here a prototypical neuron. It's like any other cell, but it has some specifics. For example, in addition to what is a cell body that would be typical for any cell, it has processes like wires. The top one is called dendrite, essentially receives information from other neurons, and then the other branches send information to other neurons. So it's a really receiving, integrating, and emitting information unit. And in the brain, I showed you a prototypical one, but there are multiple shapes of neurons. In fact, one of the founding fathers of neurobiology, Santiago Ramón y Cajal, at the beginning of the 20th century, he called them the butterflies of the soul, because he was a neurohistologist, as we call them. They studied the cells in the brain, so he was able to stain different types of neurons, and you can see they come in different shapes. Some have a triangular cell body and then long processes. Some are much shorter. Just to give you an example, in the brain there are neurons that are as short as 1 millimeter, and then you have neurons that go from the spinal cord to the foot to command the muscles there. They are 1 meter long. So they exist under multiple shapes. And very poetically, he said, as I said, he called them the butterflies of the soul, and it's really this nice quote. Like the entomologist in search of colorful butterflies, my attention has chased in the gardens of the gray matter cells which, with delicate and elegant shapes, the mysterious butterflies of the soul, whose beating of wings may one day reveal to us the secrets of our minds. It's really a nice quote. So, but these cells still, despite the fact that they have different shapes, essentially function in a similar way. As I said, they receive information in the dendrites, and then they emit signals from what are called the axon. And we have a lot of neurons in the brain, about 80 billion neurons. That's a lot. And each one is connected to more or less 10,000 other neurons through particular entities that allow this transmission of information, which are called synapses. So, which are denoted there with these red cycles. So where an axon of a neuron terminates, there is some space, and then the information is transmitted to the next neuron. So it means that if we have about 80 to 100 billion neurons, and each one is connected to about 10,000 other neurons through synapses, we have not less than 10 to the 15th synapses in the brain, meaning places at which information is transmitted from one neuron to the others. Now, how is this information transmitted? Well, through chemicals. The end of the neurons contain special chemicals called neurotransmitters that are released and then will act on receptors on the next neurons and trigger a series of response. So this, what I just gave you here in 4 or 5 slides, is really the essence of the neurobiology of neurons, and the idea that the brain functions based on the information processing of these units has been entrenched for a long time in neuroscience. But actually, there are not only neurons in the brain. There are the other half of the brain is made up of what are called glial cells. And in fact, in the human brain, these non-neuronal cells outnumber neurons. There are more glial cells than neurons. So you got to think that, you know, nature usually doesn't do or allow things to evolve in a way that, you know, they are not useful. They must do something. If half of the brain is made out of these non-neuronal cells, they must be good for something. The problem is that these cells, which, by the way, were discovered over almost 200 years ago, 150 years ago, by this German scientist called Rudolf Virchow. But he thought that these cells were some sort of glue. That's the name glia comes from. Some sort of connective tissues that keeps neurons together. And the problem with the problem, in a way, but the reality is that these glial cells, unlike neurons, do not produce electrical signals. For neurons, you can put an electrode in a neurons and you will record potentials, membrane potential, synaptic potential, action potential. There is an electrical activity. You do this with glial cells, you don't record anything. And neuroscience has been dominated by electrophysiology, and it's not a criticism, that was just the reality, but they could only see, quote unquote, neurons. They could not see glial cells because the instruments that they had were not appropriate to study these cells. And so the conclusion was, not really a conclusion, but the feeling was, well, they're probably connective tissues, they keep neurons together. But if you now take another consideration, an evolutionary consideration, I just told you that the ratio, this is in French, I apologize, means human, the ratio is 1.5, 1.6 to 1. So there are more neurons, more glial cells than neurons. But if you take the leech, or the worm, or the I'm just blanking. Frog, yeah. mice, rat, all of them have many more neurons than glial cells. So again, without being pretentious, humans probably have some more capacities or computational capacities than a leech. And maybe the fact that there are more glial cells than neurons in the human maybe has something to do with these computational capacities. So, but still, the question remained open because the tools were not available. But at least there was a distinction among these glial cells. And I will not go in details, but there are three main types of glial cells. The astrocytes, and I will tell you something about them. Then these oligodendrocytes, these are important cells because they actually produce the myelin, and myelin is the ensheathment of axons, the parts of the neurons that conduct electricity, conduct the signal. And it's like for an electrical cord, you have a plastic or rubber around it to prevent short circuits. Well, in a way, for neurons, it's the same. They have this kind of rubber, it's myelin. So these non-neuronal cells, the oligodendrocyte, produce myelin, this ensheathing layer. And then microglia are cells that are involved in inflammation and immune responses. So they're more involved in disease states. So I'll spend more time telling you about astrocytes. So these are called astrocytes. And you can, I think, appreciate why. Because they are like, their shape is like a star, astro. And here are astrocytes. You see that they are very densely packed. Each one has a certain spatial domain, which only partially overlaps with others. So they are present throughout the brain with this sort of density. Now, another interesting point, if you take an astrocyte and a neuron from a fly, this is a very schematic representation, but then you look at the human, the neuron in humans hasn't changed much in terms of, if you take a neuron from a fly or a neuron from a human, they're not very different, at least the short ones. But the astrocyte is much, much more complex in humans. Again, another question. There are more astrocytes, more glial cells. They have a more elaborate shape. Well, maybe they do something important. So what do these astrocytes do? Well, essentially, they are not only have this shape, as I said, and this density, but there is another interesting feature. You can see there where the green arrows are, that these astrocytes have a process that is close to a blood vessel. The brain is full of blood vessels. The brain uses a lot of energy. Actually, 20% of the energy of the body is used by the brain, while it's only 2% of the mass. So it uses 10 times more than its mass predicted. Yet, actually, the brain as a computational machine is pretty thrifty. It uses only 20 watt. If you think how much our Shaheen uses, well, actually, 5 megawatts. So the brain is pretty efficient energetically. And who knows, maybe it's due to what I'm going to tell you in a moment. So these astrocytes have this process that is close to the source of energy. For our body, any cell in the body, our energy comes from sugar and oxygen, which is carried around the body by the blood flow. So this already, at the very beginning, the few people who became interested in these cells were saying, well, maybe these astrocytes are important to deliver energy to neurons. So indeed, provide energy to neurons. But work from our lab and other labs have shown that actually these cells are important for memory, and they are important for protecting neurons. And when they dysfunction, possibly they are the cause of neurodegeneration. And Yves Agid will tell you more about these clinical implications of the function of these cells. Now, this is another way of showing the same, a bit more artistic. So there you have in blue, purple, the astrocyte. As you see, they have these processes around the blood vessels and other processes, and that's the other very interesting feature of astrocytes. They surround the synapses. So I showed you that the synapse is where the neurons communicate between each other. They send signals with the neurotransmitters, but these synapses are not floating around in the brain. They are actually surrounded by processes of astrocytes. And in fact, this allows astrocytes to sense whenever neurons are active locally, and they send a message. This is something that we discovered in the 90s. They send a message to the astrocyte. I mean, the astrocyte senses when neurons are active and gives information to bring in extra energy. So astrocytes act as local regulator for energy on demand. And the brain uses a lot of energy, like a muscle. When actually a muscle is active, it needs energy. And this has been at the basis of the development of these techniques that I'm sure you have seen pictures of them, functional brain imaging techniques, where you see areas of the brain lighting up when you perform a certain task. You move your hand, well, your motor cortex will be activated. I'm speaking now, my speech area in my brain is more active than when I don't speak. And all these techniques, and no point in going in the details of their name, but all these techniques, they actually do not measure the electrical activity of neurons. What they detect is actually the energy that is necessary for the electrical activity of neurons, because they detect glucose, sugar utilization, oxygen consumption, or blood flow. So this is just to show you what you can see. When you look at words, you see the posterior part of the brain more active. When you're listening, like you're doing now, you see this part of the brain that is involved with auditory function that is more active. Anything in yellow and red is active. What I'm now doing is saying words, and then you see that here my speech area is more active, and so on. So, in summary, these glial cells are performing tasks that are becoming only recently evident thanks to new technologies, to imaging, to genetics, to optical techniques that allow to look within individual cells, independent of the electrical signals, which were the only signals that scientists could detect, and that's why they only detect the activity of neurons. But in addition to the metabolic and energetic support that I showed you, which is certainly a very important function, astrocytes and probably the other glial cells are important for what is called neuronal plasticity, which is the ability of synapses to become more efficient, and they are involved in learning and memory. They are very important in clearing from the extracellular space either signals that are not wanted, or to improve the signal-to-noise ratio, and also to maintain the extracellular milieu proper for neurons. They somehow keep neurons happy, if I may summarize. And more recently, there is evidence that actually this astrocyte could even tweak, modulate the efficacy of synaptic transmission. So, in conclusions, we are just now at the beginning of understanding what these glial cells are doing, and it's not only basic science that is actually making these progresses in understanding their function, but now, thanks to work of clinicians like Yves Agid, we know that they are also involved in other higher brain functions and in diseases. And so Yves Agid will now tell you about what these basic findings bear onto higher brain functions and diseases, and pinpoint even further the importance of this forgotten half of the brain.
I won't kiss him, but I'm the co-pilot. So now we are dealing with this neuron, you see, at the bottom of this figure. This is a drawing from a drawing of Ramon y Cajal, you heard of. And you have the astrocytes just around. And you heard from Pierre that these two elements, cellular elements, are talking together all the time. And what I would like you to get in your mind, among all the properties described by Pierre Magistretti, one is of extreme importance, namely that all the neuronal afferences receive message, a myriad of messages, the type of message you are hearing presently. And the big question we have is how all these myriads of information can provide a very specific message, such as speech, movement, emotions, and things like that. And this is probably due to the presence of astrocytes. You see here 3 neurons and 1 astrocyte. And the astrocytes have this important property, namely to be able to integrate all the information from the neurons and to synchronize them. That is, all the information, all the action potential are running at the same time together on the next neurons. Integration, synchronization. That might be very important in order to explain our behavior. If, and the purpose of this short talk is precisely to see whether the astrocytes here contribute to our behavior, such as movement, I'm walking, speech, memory, etc. You certainly will accept this idea that all our behaviors originate from the functioning of neurons. If I am modifying the activity of neurons, I am modifying my behavior. If I am changing my behavior, of course, there is some change in the activity of my neurons. Everybody will probably agree with this. On the other hand, we heard from Pierre Magistretti that the neurons and the astrocytes were talking together all the time. A very important dialogue between these two cellular elements. So the question is, would it be possible that the astrocytes are playing a role, are contributing to our behavior, normal behaviors? And there are 2 possibilities. One possibility is to go through the neurons. An astrocyte gives a message to a neuron, the neuron gives a message, and your behavior is modified. But this is a provocation. Paradoxically, it may be that astrocytes might influence directly our behavior. And these are the type of questions I would like to try to answer now in 2 ways. First question, are astrocytes implicated in our behaviors? And second, are the dysfunctions of these astrocytes playing a role in various diseases of the nervous system? So very briefly, let me give you 2 examples suggesting that the astrocytes are indeed contributing to our behaviors. First, memory. This is an experiment with mice. If you inject astrocytes, you see at the bottom, an astrocytes of a mouse, in the brain of a mouse, then there is no change in its memory. But now, if you, instead of injecting the astrocytes of a mouse, if you inject human astrocytes, which are much more sophisticated, then you increase its memory. It's just an argument. This suggests simply that the astrocytes are playing a role in our capacity of learning and memory. One example. Second example, sexual preference. Romantic love, if you prefer. This is a fly, right? If you were a male fly just in front of a female fly, what would you do? You would approach the fly. You would do something we are not used humans to do. you will sniff odors, you will lick the genitals, you will mount the fly, and then have sex. That's a normal situation. Now, in this fly, if I am inducing a mutation, for instance, I introduce a mutation in the gene coding for a pheromone. You know, a pheromone is an hormone which is playing an important role in odors, and animals are, of course, very sensitive to odors. Then if you make a mutation for a pheromone called 7-tricostine, you don't mind. Then this new fly, male fly, will not only be interested by female, but also by male. Aha! This is if the mutation is an heterozygote mutation on one allele of the chromosome. But then now if you make a double mutation on the two alleles, that is what is called an homozygote mutation, then the fly becomes only interested by male. It doesn't mean that this has something to do with homosexuality, not at all. But it indicates, at least with this short example in flies, that astrocytes, again, are obviously implicated in complex mental behavior. Okay? These are two examples indicating that at least the astrocytes are indeed implicated, do contribute to behavioral activities. I'll give you a second type of example dealing with diseases of the nervous system. I give you 4 short examples. First, if you think in terms of diseases such as trisomy 21, you know that, Mongolism, you certainly heard about it, Rett syndrome, Fragile X, and if you introduce the mutation of this disease in an astrocyte, up on the left, mutated astrocytes, and if you put in a cellular culture these astrocytes with the mutation together with a neuron, the neuron will die, indicating that indeed the presence of astrocytes is obviously important for neuron to survive and eventually to die if the astrocytes is ill. One example. Second example, Alzheimer's disease. You certainly heard about Alzheimer. And astrocytes are playing a role in Alzheimer's disease in 2 ways. First, this is at the bottom, astrocytes are producing a lipid which is very important called ApoE. There are several isoforms of ApoE. If you are ApoE2, you don't get Alzheimer. You are not predisposed to Alzheimer. If you have ApoE4, another isoform of the disease, the risk is increased by 8. It's important. And is strictly dependent upon the activity of astrocytes. Second, at the upper part of this figure, in normal condition, we are all producing a protein called A-beta. A-beta. And these A-beta are destroyed by capture by the astrocytes. And normally, the A-beta is not dangerous for the brain, for the neurons. In certain conditions, in patients with Alzheimer's disease, either because there is an increased accumulation of A-beta, or because the A-beta is mutated, for instance, the A-beta 40 becomes A-beta 42, another type of molecules, then this is inducing neuronal dysfunction and neuronal death. Okay? Indicating that in this disorder, which is so common, a disaster, a sociological and political disaster for our societies, astrocytes obviously playing a role. Third example, Parkinson's disease, it's exactly the contrary, the reverse. At the upper part of, this is a work we did a long time ago in my lab. At the upper part of this figure, you see half of the brainstem. Half of the brainstem. And in black here, at the bottom, a structure called the substantia nigra. The substantia nigra contains dopaminergic neurons, and these dopaminergic neurons are damaged in Parkinson's disease. In normal brain, as shown at the upper part of this figure, the density of astrocytes is increased in the internal part of the structure on the left, whereas the density is very low on the lateral part. Interestingly enough, Parkinson's disease starts on the lateral part of the substantia nigra, as if, in other words, as if the astrocytes in normal condition were protecting against neuronal death. The reverse is observed in Parkinson's disease. As you see here in black, this is the place where the dopaminergic neurons are dying. And this is exactly also the place where the density of astrocytes is the highest, as if, again, the astrocytes were protecting against cell death. In the first case, normal condition, the astrocytes are protecting the neurons. In the second, they are trying to help the neurons to survive. In this case, in Parkinson's disease, therefore, a third example showing that in this disorder, the astrocytes probably are protective instead of deleterious. Finally, epilepsy, another example. In epilepsy, we have a very common disorder called temporal epilepsy. When you're an epileptic patient, you can have a sort of coma all of a sudden, but many patients have behavioral abnormalities. This is called temporal epilepsy. And in temporal epilepsy, it's characterized by the presence of an accumulation of astrocytes in the hippocampus, here in the temporal cortex. And in this disorder, we don't really understand what is happening. In this experiment, made in sections of hippocampus, you can activate the astrocyte on the left, activate. The astrocyte is releasing a neurotransmitter called glutamate, very high concentration of glutamate, and this is activating the neurons which become epileptic. In other words, they don't have normal action potential like that, but an epileptic discharge, like that, which leads to the coma of patients usually. But interestingly enough, in this experiment, if you block the activity of the neurons, using tetrodotoxin, for those who are neurobiologists, then, in contrast to what was expected, that's science. The activation of the astrocytes can induce the epileptic discharge. In this case, the astrocytes is providing the disease without neurons. These are few examples. Of course, I could give you many more examples. So I would like to terminate before being at your disposal for questions, together with Pierre, by two conclusions. The first conclusions deal with pharmacology. You know, in psychiatry and in neurology, we have many drugs in order to alleviate symptoms. Antidepressant, anxiolytic, drugs against schizophrenia, Parkinson's disease, etc. But we have no way, we don't know how to cure the diseases. None of them. It's like aging. In contrast to what happened for cancer, for heart disease, for pancreas, infection, etc., etc. In particular, in the field of neurodegenerative disease, such as Alzheimer, Parkinson, lateral sclerosis, cerebral ataxia, etc., we don't know how to treat, to cure the patients and how to prevent. So we need to find new drugs. I'll give you an example. In the field of Alzheimer, during the last 10 years, there have been 10,000 different trials with drugs. And no results whatsoever, except in one case, a drug that is, in fact, useless when I don't use it because it's too weak. So it's a real massive problem for us. And the reason for that, I think, is that our colleagues from industrial company are only trying to find drugs targeting the neurons, receptors on the neurons, membrane on the neurons, but never on glial cells. So what I would suggest, will suggest for the future, indeed, is to try to synthesize drugs which would not only act on the neurons, but also on the astrocytes, hoping that this will be helpful. That's the first conclusion. The second conclusion is related to cognitive science. Why do we think? What is thought? What are our beliefs, etc.? All the models of the functioning of the brain have been so far dedicated to the study of neurons, exclusively. The models by Edelman, the models by Linas, the models by Changeux, and many others, all are focusing, are dedicated to the study of neurons. Even the Human Brain Project. I was in the scientific council of this project. Nothing about the astrocytes. Nothing about the other half of the brain. How is it possible? So my feeling is that if you see an astrocyte on the left and the neuron on the right, it's a female, an astrocyte, and a male, the neuron, but it could be the reverse. It's just a joke, right? We are, therefore, we think, and in the future, Pierre and I, really are hoping that all the models of the functioning of the brain, which might explain our motor activity, our intellect, our emotions, will be based on the physiology not only of neurons, but also of glial cells. Well, thank you for your attention, and I think we are at your disposal if you have any questions.
Questions? Yes. Okay, maybe stand and speak up. I don't know if there are mics. Sorry? Oh, here. Okay. The box. Okay. Wow.
You can throw it like this. No? Ouais,
Fantastic.
pas mal. Super.
Thank you.
I'll get the trick for the future.
That's nice.
Thanks for the talk. Can you guys please elaborate on the role astrocytes and even neurons play in memory, like memory formation and storage, I guess, in humans? Mm-hmm. Thanks.
I can give you one answer which is based on our work, and then maybe Yves can elaborate also. What we when I showed you this, what we call neuron-astrocyte metabolic coupling, you know, I showed you that astrocyte can sense when neurons are active, and they import glucose from the blood. And then the question you may ask, okay, but what do they deliver in terms of energy back to the neurons? And we made an observation which was quite controversial at the beginning, because what we found is that what the astrocyte delivers to neurons is a molecule that was long thought to be actually a metabolic end product, not a useless end product, called lactic acid. And I'm sure you have heard about it. Those who run, you know, they know that lactic acid can accumulate because there is not enough oxygen to burn the glucose in the muscles. And so, in general, lactate was considered to be a waste product because of lack of oxygen or insufficient oxygen. But actually, it was only a partial view. There were evidences that lactate can be formed even when there is enough oxygen. So what we showed is that this astrocyte takes up glucose from the blood, produces lactate, and the idea was that then this lactate can be gives it to neurons, and then it can be converted to other metabolites like pyruvate, it's a bit techy, and then burned and produce energy. That was one, but that is not directly related to memory. What we showed with a colleague of mine in New York at NYU, Christine Alberini, is that if we block the transfer of lactate from astrocytes to neurons, we actually block memory. And we can rescue this if we administer locally in this part of the brain involved with this particular type of memory called the hippocampus, we can rescue. So suddenly it became obvious that this transfer of lactate from astrocytes to neurons is involved in memory. But the next question was, is it energy? Of course, if you don't have energy, you cannot store memories. But it was not energy. It was something special that this molecule does, which is to modulate a process that is called neuronal plasticity, which is at the basis of memory. So lactate is not only energy, it's also a signal for plasticity and hence for memory. So this is an example in which we now have a molecular basis of how astrocytes are important for memory consolidation. But also you gave another example with a transplant. And this is, I don't think it's known exactly why, if you put these more elaborate astrocytes into a mouse.
Yes, but there are several other experiments. Are you satisfied with the answer, or would you like me to talk a little bit about the problem of memory? What is memory?
Yeah, I'm not sure how even neurons affect memory in the first place, not just astrocytes.
Yeah. Okay, well, the idea is essentially that these 10 to the 15th synapses that the brain, that neurons are connected, they don't transmit information at the same intensity and efficiency all the time. When a group of neurons is engaged in a learning, in a behavioral task that eventually will lead to learning, there are molecular changes that occur at the level of certain number of synapses that make them more efficient. So actually, the basis of, at least one of the basis of memory is that a certain number, maybe thousands of synapses that has been strongly engaged in a behavior become facilitated. And so when they are reactivated, they are more efficient. And astrocytes play a role in allowing this facilitation.
For me, the real problem with memory is what is memory? How our brain, which is a material piece of work, it's a material. I have a brain, it's almost 1.5 kilos, with all these neurons and astrocytes, glial cells. But how this brain is able to produce our thoughts, thinking, which is immaterial. We don't know what it is, what is thought. And that's the problem. At the cellular level, we have a lot of information. But what about, I mean, at the physiological level? That's still a big mystery. And in that sense, to study the respective role of astrocytes and neurons is very difficult. Why? Because, I'll give you an example. Our brain is connected to the environment. It's a machine. It's a machine connected to the environment. I perceive, I am acting, and I am processing a lot of information in my brain in order to produce my actions. I see you, I receive this information, just what you said, and now I try to answer a question. And in between, my brain is activated. When I received your information through my ears, this information became electric current in order to reach my my auditory cortex in that sense. When I speak with my mouth, with my tongue, with my larynx, with my gesture, then I'm using my neurons again, without astrocytes. So each time I perceive, each time I'm acting, it's only neurons, electric currents on the neurons. You agree with that? But inside the brain, all the processes of the formation of our thoughts, emotion, intellect, strategy of action, perception, language, etc. All this includes the activity of not only neurons, but also astrocytes. But how can you evaluate this? Because obviously you need to use your peripheral nerves, those who give you the information and those who are activated, who provide your actions. So it's very difficult to see what happened in the brain. Probably in the next future, several methods will be able to activate differently the astrocytes and the neuron. Then I will be able to probably answer more clearly your question. Up to today, it's not possible. But in the near future, I think we will be able to tell. Yes, please.
Okay.
You want the ball or?
Is it true that the human heart can also store some information in terms of memory?
I can easily answer the second question. The answer is no. The heart is simply a pump. But by tradition, in your country and in my country, we always say that our emotions are in the heart. If you feel in love with someone, sir, then you will do this. You don't do this. Okay. There is for sure for that. But, so the heart is simply a pump. We need it, of course. But all our emotion and memory comes from here. So the amount of capacities of memory of our brain is extremely difficult to evaluate. Your question is extremely complex for me. Perhaps you would better answer that question. The capacity of the computers in terms of memory are much higher than ours. The problem with us is that we have this brain plasticity, and we can change our memory. In fact, during life, since you were a small boy, and until now, when you will be an old person, a given memory will change its significance with time. You won't get exactly the same. So, and at the end, as you know, like I do myself, I am starting to not to memorize the name, the family name of my friends eventually, so that's a big problem. So the memories of the computers are much more efficient than our memory. But it's less.
I don't know if it's efficient, but they are certainly much larger. But also, I think
and quicker too.
The efficiency of our brain is probably in the processing, which is most likely not a binary one. And so then the issue of memory is different than in a binary system.
That's where perhaps the other cells of the brain, we see, Pierre and I, At difference from several scientists, by the way, that the neurons are simply wires, electric wires, which are there just to run all the information through the brain, even in a very complex manner. But the astrocytes may play a role there. That's a fantastic avenue of research for the future for us.
Excuse me.
Here, maybe the little box. Can we send it here?
Wow!
Yes,
Pas mal.
well done.
Well done. Well played.
Yes, is it true that any moment throughout all our life is stored in our brain somewhere, even if we don't remember them, and by somehow it can be retrieved or triggered again?
This is it's a very interesting question. There are clearly we have consciousness and awareness of reality, but the view more and more is that it's like the tip of the iceberg, our conscious experience, that we have a very large component of our mental life which is not conscious. Now, it can become conscious very easily in certain cases, but in others, not. It's a very controversial field. I think there is, in general, an agreement. The image of the iceberg, I think, is generally agreed upon, so that we think we are in charge of our life because of our conscious experience, but in fact, we have the major part of the iceberg underwater or under consciousness, and that part also influences our behavior. And it's a bit distressing in a way, because it would mean that we are not necessarily, we don't in charge of everything we do or feel. And I think this is true. So this aspect of the unconscious has been explored very deeply by the psychoanalytical theory of Freud, but even without considering the psychoanalytical theory, I think there is in general agreement that we have an important component of our life that is non-conscious.
We know a lot about the physiology of memory. When you learn something, you have 3 steps. First, you encode, for instance, the word, I don't know, a pen, for instance, a pen like this. You want to memorize it. It needs to be encoded. Then it's stored somewhere in the brain. And then you have to recall, what is this? So the information is somewhere in your brain, and you have to recall. 3 steps: encoding, storage, and then recall. We know the structure which encode. This is a temporal lobe here, and a small structure called the hippocampus. We know it perfectly well. And this is, by the way, the brain structure which is first damaged in patients with Alzheimer. As you know, patients with Alzheimer start to lose their memory. They start to lose their capacity of encoding. Then, when you have encoded this information in the hippocampus, then it's stored somewhere. For instance, a pen can be described by its form, its color, its topography. Here, it's 3 index. And that is stored in the cerebral cortex. That's in the cerebral cortex. The form in some places, the color here, the topography here, for instance. We know roughly what happens. So that when you recall in information, if you ask me, do you recall the pen? I have to, then in my brain, what happened? All the information, the color, the form, the topography, goes to the hippocampus. Then this is a place where the information is located, and then I can recall it. So it's, we start really to understand the physiology of memory, if that was your question. Yes,
I think we have time for a last question. I think here. One or two, maybe. One, and then. Can you raise your hand? Who wants the ball?
Formidable. You're very good, huh?
Oh, thank you. Thank you, professors. That was very impressive. My question is, why do you think, from your point of view, people have just concentrated to measuring neuronal functions and I don't want to say like not studying glial cells? Is it that hard to measure a function of a glial cell? Neurons, we can measure the action potential, as you said. But a glial cell, how can I see it? You remember you said at the beginning it's not easy to see these cells. Is it technically difficult? Thank you.
Well, I can start and then Yves can continue. Yes, it's really, certainly, there is a technology or technical reason. As I said earlier, neurons produce electrical signals. So if you have an electrode, you can record electrical activity, and then you can analyze the encoding, the pattern of activity, and all that. Glial cells do not produce electrical signals, and that was the only tool that actually neurobiologists had. And then in the 80s and 90s came very high resolution imaging techniques where you can use dyes that can be taken up only by glial cells, and then you can see that these cells become very active when neurons around are active. So suddenly it was possible to see glial cells. Also, there techniques evolved in which you could separate astrocytes and neurons and study them separately and together. So certainly there is a technical reasons. There may also be some ideological reasons why, but maybe Yves, you can elaborate on that.
Since I am your co-pilot, yes, there are also historical reasons. Neurons and glial cells were initially described by a Frenchman, by the way, Mr. Dutrochet, in 1824. Virchow was 1856. Although Virchow was made an excellent description. But at that time, until the 1920s, both astrocytes and neurons were described together by Mr. Ramon y Cajal, by Golgi, by Andriessen, by Rio L. Ortega, and many others, by Charcot and others. But in 1920, suddenly, there was a possibility to record the neurons. When you put the electrode on the neurons, you are recording an action potential. And this was the explosion of discoveries until today. Fortunately, we still use this method. That is to study exclusively the neurons, because there was the astrocytes are mute. They don't speak. Therefore, the only way to study these cellular elements at that time was morphology, histology, through histology using microscopes. And between 1920 and 1950, there was almost no research in the field, up to the 1950s, where new methods allowed to study these elements again.
We have time for a very quick question, I saw, but we are getting actually up to the time.
I'm sorry, I don't hear you. You speak loudly, or you...
Where
is the ball?
Can you send the box?
It's finished?
No, just very last. 30 seconds.
Thank you for the presentation. May I know some ways in your life that you keep your memory in good condition, not forget any data sometimes in your life? What ways you can, do it in your life to keep your memory in good conditions?
To improve memory?
Yeah.
There is no drug.
No.
There is no drug. Be careful, it might be dangerous. There is no drug, almost no drug. So in patients with Alzheimer, we give some drug which induce perhaps a 5% improvement during a short period of time. So the best way to improve your memory, like you and me, is to use our memory. is to use
Healthy
it
life,
simply.
exercise.
Exercise,
Healthy
like
food.
doing some motor exercise to, you know, to become in good
Because
shape. So you need to become in good shape intellectually, emotionally, sexually, and from a motor point of view.